All right, everyone, here we go. Part 3 of our Muscle 2 lecture of the physiology of the neuromuscular junction. This is where we're going to put it all together. Muscle 1 may have been a little bit difficult for some of us, a little bit abstract, talking about things like voltage-gated channels and ligand-gated channels, all these ions moving, why are we changing the polarity and all that kind of stuff. Muscle 2, segment 2, that became a little bit easier to understand, but still not quite seeing, all right, where are we going with this? In this third segment, that's where we're going to put it all together. And so we're going to take a look at how we take that electrical signal and how we produce a mechanical outcome. Where we're going to pick up is where we left off last time. We looked here at the neuromuscular junction. And what I want to do is I want to minimize that and I want to maximize what's happening in the skeletal muscle fiber. We'll recreate that image here with our motor neuron. We also have our synaptic vesicles with acetylcholine. We have our acetylcholine receptors on the postsynaptic membrane, or what we call the motor end plate. We have our voltage-gated calcium channels, and we're ready to go to work. As we know, that action potential is going to propagate down the axon of the motor neuron. We're picking up again right where we left off before. As it arrives at the presynaptic terminal, that causes the opening of the voltage-gated calcium channels. Calcium will diffuse in. That causes the synaptic vesicles to fuse with their membrane, releasing their neurotransmitter acetylcholine into the synaptic cleft via exocytosis. The acetylcholine will diffuse across the cleft and bind its receptor on the postsynaptic membrane. At that time, this is where, if I were you, I would discuss all the steps of the action potential. As we know, that receptor is actually a ligand-gated sodium channel. So we have high concentration of sodium on the outside, low concentration inside, opposite for potassium. But when we open up that ligand-gated sodium channel, as we already know, the sodium will start to leak in, carrying positive charge with it. That produces a stepwise progression to a more slightly positive value. The membrane potential moves from negative 70 to negative 69, and we call that a local depolarization. As more and more acetylcholine binds to more and more of its receptor sites, we open more and more ligand-gated sodium channels. More and more sodium will leak in, carrying positive charge with it. We produce more local depolarizations. The membrane potential will then continue to move from negative 69 to negative 68 to negative 67, negative 66, and so on. If we get enough acetylcholine binding to open up enough ligand-gated sodium channels to have enough sodium leak in to produce enough local depolarizations, we will hit threshold. At negative 65 millivolts, that's what threshold is. Voltage-gated sodium channels open, sodium having a high concentration outside, low concentration inside. We get this huge influx of sodium carrying positive charge with it. Because we're flooding the cell with positive charge, the membrane potential skyrockets to a plus 30. We call that a rapid depolarization. At plus 30, the voltage-gated sodium channels close, voltage-gated potassium channels open. Potassium having a high concentration inside, low concentration outside, we get a huge efflux of potassium, carrying positive charge with it. The membrane potential will plummet back down to a negative 70 millivolts. We call that a rapid repolarization. At negative 70 millivolts, the voltage-gated potassium channels close and the sodium-potassium pumps reestablish their concentration gradients. Meanwhile, what we've done is we've generated an action potential right here in the skeletal muscle fiber, right there at the neuromuscular junction, what we call the motor end plate. That action potential now is going to propagate down the sarcolemma of the skeletal muscle fiber shown by the arrows here in red. As the action potential propagates, keep in mind that is a wave of positive charge. So as I draw another arrow here, that's converting this area of the sarcolemma to a plus 30. Meanwhile, don't forget, behind that action potential, we're now doing repolarization. That's where the voltage-gated potassium channels have opened. Potassium is now leaving the cell. Then we're going to plummet back down to the negative 70. We're kind of hitting the reset button. Meanwhile, what happens is this action potential is going to continue to propagate down the sarcolemma until it reaches the T-tubule. The T-tubule will take the sarcolemma depolarization down deep into the sarcoplasm, where it will interact with this specialized organelle we call the sarcoplasmic reticulum. You'll remember from Muscles 1, sarcoplasmic reticulum is a specialized smoothie R. It's specialized because it stores a lot of calcium, 2,000 times as much calcium as in normal smoothie R. Do you think calcium is going to be important for muscle contraction? Yeah, you bet it is. What happens is that when that action potential, that wave of plus 30 right here shown by these arrows, hits the sarcoplasmic reticulum, it opens up voltage-gated calcium channels in the SR, and massive amounts of calcium will flood the sarcoplasm. Remember this guy? That's our sarcomere. Myosin in red, actin in black. The only thing we've introduced is what's shown here in pink. That's a regulatory protein called troponin. When the sarcomere is at rest, that troponin physically blocks the myosin binding site on actin. So therefore, it's like having a physical barrier here. The myosin heads cannot contact the actin. But when the sarcoplasmic reticulum opens up its voltage-gated calcium channels and calcium floods the sarcoplasm, calcium is actually used to bind onto that troponin. Once it binds onto the troponin, it changes the conformation of troponin. Troponin rolls out of the way. And now for the first time, the myosin binding site on actin is now exposed. Guess what? We're almost there, folks. We are ready for muscle contraction. Through a first step we call cross bridge, the myosin heads latch onto the actin. They are now physically linked. Through a second step that we call power stroke, these myosin heads, which are already in the cocked position, they're like a set mousetrap, they snap forward, and because they're physically attached to the actin, they pull the actin over the top of it, and the bare zone narrows. Through a third step called recovery stroke, ATP is used to dislodge the myosin heads from the actin, but then it recocks those head groups so they can reattach. Through many quick repeats of the cycle, through many quick repeats of cross-bridge power stroke and recovery stroke, what happens is the myosin heads will walk right down the actin, pulling the actin over the top of itself, and that bare zone eventually disappears. Once it eventually disappears, we are at full contraction. How do we relax? When there is no more signal present, that means there's no more action potentials here. That is a signal for ATP to dislodge the myosin from the actin and to pump calcium back inside its storage containers in the sarcoplasmic reticulum. That requires active transport. When that happens, and we get all that calcium out of the sarcoplasm, and again, the myosin heads are dislodged from the actin, then the sarcomeres passively lengthen, which means the myofibrils passively lengthen, and our skeletal muscle fiber passively lengthens, and now we are at full relaxation. So that is our take-home message. That's the story that we want to be able to tell. We've used these three parts of Muscle 2 lecture to be able to tell that story. That puts it all together. Puts all the steps of the action potential together. It puts all the steps of the neuromuscular junction, and finally puts all the steps of what's happening inside the skeletal muscle fiber. Again, we've taken an electrical signal and now we produce a mechanical outcome. By the way, this is also what I want you to do for your next review assignment. You notice from our syllabus, we have 2 review assignments, each worth 10 points. The first one was on chemistry. This is your second one, the physiology of the neuromuscular junction. What I want you to do is I want you to start with the diagram in front of you. By the way, I like this diagram a lot better than the figures out of your textbook, but you can use the figures out of your textbook. They're nice. They'll look like this, and they'll take you right through the steps as well. But if you use my diagram, it's a simplified diagram, and I want you to start with that. So starting with your diagram of this, again, your own drawing. Of course, you're going to model it after my drawing, but I will not accept just a printout of what you see on the screen in front of you. I want you to actually draw this yourself. And then I want you to go step by step by step through every process and every step that we just described. That's what you're going to submit to me as part of your review assignment. By having you do this yourself, my hope is that you'll increase your understanding and your retention, because I want you to do the drawing and I also want you to memorize all those steps. That's going to get you in good shape for our next quiz as well as our exam. So again, one more time, let's go through those steps. Starting with the action potential propagating down the axon of our motor neuron, it arrives at the presynaptic terminal. As it arrives at the presynaptic terminal, it opens up the voltage-gated calcium channels. Calcium will then diffuse in. As calcium diffuses in, it causes the synaptic vesicles to fuse with their membrane, releasing their neurotransmitter acetylcholine into the synaptic cleft via exocytosis. The acetylcholine will diffuse across the cleft and bind its receptor on the postsynaptic membrane. That receptor is actually a ligand-gated sodium channel. When acetylcholine binds its receptor, the ligand-gated sodium channel opens. Sodium having a high concentration outside, low concentration inside, sodium will start to leak in. As it starts to leak in, it carries positive charge with it and produces what we call local depolarization. Again, folks, this is where I would discuss every step of the action potential. Taken right from the first segment on Muscles 2, that first video, write them all down. What happens is, as more and more ligand binds onto more and more of its receptor sites, we open to more and more ligand-gated sodium channels. More sodium will leak in, producing further local depolarizations. The membrane potential moves from -70 to -69 to -68, -67, and so on. If we get enough ligand, that is enough acetylcholine binding onto its receptor sites to produce enough local depolarizations, we'll hit threshold. At -65 millivolts, that's the signal to open up the voltage-gated sodium channels. Because we have a high concentration of sodium on the outside, low concentration on the inside, sodium rushes into the cell. We say we get a huge influx of sodium, carrying positive charge with it. the membrane potential skyrockets to a plus 30. We call that a rapid depolarization. At plus 30, that's a signal for the voltage-gated sodium channels to close and the voltage-gated potassium channels to open. Potassium having a high concentration inside, low concentration outside. Potassium rushes out of the cell. We call that a huge efflux of potassium carrying positive charge with it. The membrane potential plummets back down to a negative 70 millivolts. At negative 70 millivolts, that's a signal for the voltage-gated potassium channels to close, and the sodium-potassium pumps reestablish the concentration gradients. Meanwhile, we've established an action potential in the skeletal muscle fiber. That action potential will propagate down the sarcolemma of the skeletal muscle fiber until it reaches the T-tubules. The T-tubule will take the sarcolemma depolarization down deep into the sarcoplasm until it reaches the sarcoplasmic reticulum. Once the action potentials reach the sarcoplasmic reticulum, it opens up voltage-gated calcium channels, and massive amounts of calcium will flood the sarcoplasm. The calcium will then bind onto a regulatory protein called troponin. Troponin then will change its conformation and moves out of the way, and for the first time, the myosin binding sites on actin are now exposed. With the myosin sites on actin now exposed, we are ready for contraction. Through a first step called cross bridge, the myosin heads latch onto the actin. The two are now physically linked. Through a second step called power stroke, the myosin heads, which are already in the cocked position, will pivot, pulling the actin over the top of itself, and the bare zone narrows. Through a third step called recovery stroke, the myosin, ATP is used to dislodge the myosin heads. The myosin heads then will become recocked, and through many quick repeats of the cycle, the myosin heads walk down the actin, pulling the actin over the top of itself, and the bare zone narrows until it disappears. Once it disappears, we are at full contraction. When there is no more signal present, that means there are no more action potentials. Then ATP is used to dislodge from the myosin from the actin and to pump calcium back inside its storage containers in the sarcoplasmic reticulum. The sarcomere passively lengthens, which means then the myofibril passively lengthens as well as our skeletal muscle fiber, and now we are at full relaxation. So that's our story. That's what we want to be able to practice. That's what we want to be able to tell. We've got our diagrams here, these drawings. Again, I prefer those over the textbook, but you can rely on the textbook as well. But you've got all these drawings. You've also got my verbal explanation here on this video. In addition to that, we've got it in the lecture notes. So let's do a little housekeeping like we've done with the first two segments. Start on page 84. Let's look here at excitation, contraction, coupling. Excitation contraction coupling is a sequence of events by which transmission of action potential along the sarcolemma leads to the sliding of the actin and myosin myofilaments. The action potential is brief and ends well before any signs of contraction are obvious. The action potential also does not act directly on the myofilaments and instead causes the necessary rise in intracellular calcium ion concentration that is required in order for actin and myosin myofilaments to interact and slide. Top of page 85, the sequence. The action potential that was initiated at the neuromuscular junction propagates along the sarcolemma until it reaches the T-tubules. The T-tubule imagination takes the sarcolemma depolarization into the sarcoplasmic reticulum. Voltage change, that is the depolarization to plus 30, in the T-tubules causes the voltage-gated calcium channels in the sarcoplasmic reticulum to open and massive amounts of calcium diffuse into the sarcoplasm, all within a millisecond. Calcium then binds onto the regulatory protein called troponin, changing its conformation and exposing the myosin binding site on actin for the first time. With the myosin binding site on actin now open, myosin binds onto the actin through a step called cross bridge, and contraction begins. 3 steps. Cross bridge. The myosin heads bind actin, and actin and myosin are now physically linked. Power stroke. The myosin head pivots and bends, pulling the actin over the top of itself, and the bare zone narrows. Recovery stroke, which is the cocking of the myosin head. At the cost of ATP, the myosin head is dislodged from the actin and returns to its pre-stroke high energy or cocked position. If calcium is still available because of continued stimulation, the myosin head engages. Therefore, through many quick repeats of the cycle, the myosin heads are literally walking along the actin filaments, much like a centipede's gait, and the sarcomere shortens and contraction is achieved in each fiber that has been stimulated by the neuron. The actin filaments cannot slide backward as the cycle repeats again and again because some of the myosin heads, the legs that is, are always in contact with the actin, which would be the ground. Flip the page over, top of page 86. It is likely that only half of the myosin heads of a thick filament are pulling at the same instant. The others are randomly seeking their next binding site. Relaxation. If no more stimulus is present, that means there is no more action potential, then calcium ions are rapidly pumped into the sarcoplasmic reticulum by active transport. Yes, that does require ATP. ATP is used to dislodge the myosin from the actin and recock the myosin heads. With free intercellular calcium no longer present, troponin returns back to its original conformation and blocks the myosin binding sites on actin, and the sarcomere passively lengthens. So again, we've got several different sources by which to use to come up with a good understanding of this process. You are absolutely going to see this, as you know, on your next quiz and exam. So as we've done with other physiological topics, what you want to do is be able to practice telling the story. Again, especially in this class, I think a figure like this is worth 1,000 words. So start with a figure like this, not only for your review assignment, but also for your study and preparation. And then memorize all those different steps. If you start with a figure like this, you can take yourself right through this, and you want to practice telling the story. Practice telling your story to someone or something, your houseplant, your dog, your cat, a loved one, it doesn't matter. As soon as you get comfortable telling this story, that means you've got it, and you're going to be in good shape for our upcoming quiz and tests here in not too long. Before we finish up with rigor mortis, our final clinical example here for muscles 2, let's just remind ourselves, again, do you think calcium homeostasis is important inside the body? Yes. Do you think muscle contraction requires a lot of energy? Yes. Let's look at those two things. Remember, when we talked about the skeletal system, calcium homeostasis is absolutely essential because calcium, though, has to be available for 3 and many more actually important processes. 2 of those you see right here on the screen in front of you. One, of course, is nerve cell communication. Again, if we don't have enough free calcium, we cannot we cannot diffuse into the presynaptic terminals. We'll never get release of our neurotransmitters, so nerve cell communication stops. Also, if we don't have enough calcium, then we can't bind onto the regulatory protein called troponin, and therefore we can never open up those myosin binding sites. Again, contraction stops. We also, by the way, need calcium in cardiac muscle too. In addition, not only to calcium being very important, again, muscle contraction uses a lot of energy, a tremendous amount of ATP. In fact, it uses so much, we've got to be able to produce it as fast as we're using it in most situations. As I look at the screen in front of us, I see the ATP being used in 3 different ways. Number 1, our sodium-potassium pumps. These pumps are always on. When we open up the voltage-gated channels, whether it's sodium or potassium, we can overwhelm the pumps, but the pumps are always on, so we're always using a lot of energy when it comes to that. Secondly, as we talked about, when we perform the recovery stroke, ATP is needed to dislodge the myosin heads from the actin and to recock that. Because we go through many quick repeats of this cycle in order for muscle contraction to occur, we're burning a ton of ATP very fast. So again, oftentimes in these skeletal muscle fibers, we've got to be able to generate that ATP as fast as we're using it. Third, we also are going to need ATP to pump calcium back inside its storage containers inside the SR. So therefore, if we don't have ATP, we can never pump the calcium back into the sarcoplasmic reticulum. If we cannot pump the calcium back in the sarcoplasmic reticulum, are we going to get relaxation? No, we're not. In fact, that's the basis for our third and final clinical example here, rigor mortis, page 86. With rigor mortis, what happens, most muscles begin to stiffen within a few hours after death, with a peak rigidity about 12 hours, and then it'll gradually dissipate over the next couple of days. The reason why it gradually dissipates is not because all of a sudden the cell came back to life, but instead the proteins here start to break down, and that's why we lose the contact between the actin and myosin. The problem is, though, is that dying cells are unable to exclude calcium, and therefore that influx in the muscle cells promotes the formation of the cross bridges. Remember, these heads are already in the cocked position. So if calcium is cruising in and binds troponin, we just open up the binding sites. These myosin heads can latch on. Because they're already in the cocked position, guess what? Power stroke can also occur. However, with no ATP, because the cell is now dead, it's the recovery stroke that cannot occur. So what happens, cross-bridge detachment is impossible. The actin and myosin therefore become irreversible cross-linked. That produces the stiffness of rigor mortis, and then again, over time, it'll gradually disappear, however, due to the breakdown of the proteins following death. With that said, folks, congratulations. We finally made it through Muscles 2. We had 3 different videos. Again, we started out a little bit more abstract, got a little bit easier to understand, though, in Muscles 2. The segment of Muscles 2, Part 2. And then in this third part, hopefully, we brought it all together. As usual, guys, if you have any questions at all, please don't hesitate to reach out. Hopefully the diagram you see before you, hopefully my narration, though, will provide the necessary steps and understanding, but I'm always here if you guys need anything. Otherwise, good news is only one more lecture, Muscles 3 coming up, and then we're ready for our next quiz and exam. You guys take care, and I'll be in touch soon. Thanks.